session. So it's a double summary. I'll start with a quote from Albert Einstein, which is good for... Remember, Albert Einstein was a genius, and he could think of all types of complicated matters and be able to create a clear path through confusion. And he came up with this statement of all people: make everything as simple as possible, but not simpler. So obviously he was not only a great researcher and innovator, but a good teacher. Today's session, and then tell you how to summarize. We have different types of articles, and all of these articles in all of the journals in the world generally follow a protocol that's set up by international standards. And this includes the Indian Journal of Ophthalmology, the American Journal of Ophthalmology, the Archives of Ophthalmology, Nature Science, New England Journal of Medicine, etc., etc., etc. In this case, I'm going to break my talk into various categories. Different types of articles that are published. Laboratory experiments that are just taking place in a lab. Next stage would be animal trials, and then clinical trials with humans. And finally, the outcome measurements. Articles, as mentioned earlier by Dr. Gupta, start with an abstract. It follows the title, and the title should be limited so that it grabs the interest of the reader. It goes to the core of the report, the purpose of the article, the hypothesis it plans to prove or disapprove, its findings, and how useful it is. And also it emphasizes the importance of these results or the lack of results. And it gives the scientific contribution and structure to the paper. That's the abstract. The introduction, as discussed by Dr. Gupta, discusses the materials and methods used for that investigation and references to similar investigations and findings. It gives a focus to the paper. It brings the reader up to date with some of the knowledge that has been distributed earlier, so that someone that's not so familiar with the topic will learn about what has developed prior to this paper being published. And the results have to present the data without interpretation. The facts, just the facts, nothing but the facts. And at the end, the discussion is more liberal, because it interprets the data from your studies and compares it with similar investigations that previously reported only in peer-reviewed journals. And finally, the summary, the conclusion. That's my topic, to summarize. And this emphasizes the significance of the report by highlighting the data derived by this investigation that may or may not support the hypothesis made at the beginning. And this summary could be a category of the abstract. Sometimes when you're reading an abstract, it breaks it into different categories, and at the end of the abstract there's a category called summary. Another area for that summary is in the discussion. Now I'll give some examples. Basic research, let's go into that. Pharmaceuticals. Okay. I'm going to concentrate mostly on the eye. Ophthalmology journals, and just like the previous speakers, imparted some additional information, not only about how to write a paper, but some good tidbits about ophthalmology. In this case, we're going to go into biochemical and pharmaceutical research. And we'll talk about antibiotic sensitivity. And I highlighted the important areas. Usually, articles talk about buildup of resistance from antibiotics, but very little was done about how there could be a crossover of collateral sensitivity from genetic changes. And so this was in the abstract, using large-scale experiments with E. coli, that there's collateral sensitivity. That's the abstract. They set out what they want to do. In the summary, it talks about large-scale laboratory evolution experiments, genetic adaptation to the aminoglycosides, and whole genome sequencing in the laboratory. Please understand that this talk is tilted towards genetics and stem cells because that's what I'm interested in. Just like the previous speakers really tilted it towards subjects that they were interested in. So in this summary, it's good. It just talks about the results of their lab experiments, showing what they set out to prove. Let's go into genetics. Again, it's laboratory. And these are a paper about electrically assisted ocular gene therapy, electron transfer iontophoresis. They wanted to see in the laboratory which method is good for transinfection other than using AAV, associated adenoviruses. And so it talks about, in the summary almost, in this abstract, iontophoresis has been found efficient for small nucleic acid fragments, and electron transfer, on the other hand, is for the large one, oligonucleotides and plasmids. So in this case, the abstract, in a way, had the summary. But in the conclusion, or another summary, it says that iontophoresis is more appropriate for the delivery. It says the same thing. So in this case, the conclusion really reiterates what was in the abstract. And it goes on to saying that this I take affront to. It's a guess. They're talking in the summary. They're guessing. Well, they shouldn't put that in there as a guess, because we want facts, only the facts in the summary. All right, let's go to stem cells, induced and redirected. This is isolation of amniotic stem cell lines with potential for therapy. And it talks about, in the abstract, stem cells are capable of differentiating into multiple lineages, and that cloning human lines, verified retroviral markings, etc., etc. This is what they set out to do. And in the summary, they conclude that, yes, it can be done. And in the summary, it makes a statement, I guess it's okay. In the future, banking of these stem cells may provide a convenient source for both analogous therapy in later life and matching. So we're talking about what they resulted. These are set as facts. It shouldn't be theory, and theory should only take place, opinion should only take place in the discussion. Now we're going into clinical trials, clinical trial type of papers. Collation of data, different phases, 1 to 3, of developed products prior to Food and Drug Administration approval, which has some impact around the world. So here, this is phase 1 clinical trials of recombinant human endostatin. Well, it gives the purpose in the abstract that they're injecting recombinant human endostatin intravenous to see how it does with refractory solid tumors. And in the summary, it says that there were no clinically significant toxicities. Okay, that was from their investigations. We'll go into collation of data after FDA approval. Once something is on the market, and this is putting together a mass of information. And you know that there are many drugs that have been taken off the market because of effects that were seen only after millions of people had taken the drug, which did not come out during the phase 1 to 3 trials. So anyway, here we have an abstract about lutein and xanthine and omega-3 fatty acids for age-related macular degeneration, the ARIDS 2 study. And it was a question of whether adding 2 types of drugs to the ARIDS formula, lutein and xanthine, omega-3s, and EPA might further reduce the risk of age-related macular degeneration. Is it significant? Does it do anything? So the abstract in effect asks that question. It does not give an answer in this case. But take a look at the summary. The addition of lutein and xanthine and DHA, various fats, to the ARIDS formula, either the lutein and xanthine and or the fats to the formula, in the study, millions of people, did not further the risk of progressive to advanced AMD. Okay, now that's significant. It was negative. Comparison of low dose zinc versus high dose zinc showed no evidence of significant effect, and there's insufficient evidence to provide a clinical recommendation. The only thing is that the lutein can replace the beta-carotene that has some adverse effects, I believe, on the heart. So that's about the one thing that came out of that paper. So, in summary of my summary and summary of this session, I'll say that the summary should briefly highlight the important conclusions of the reported research or investigation. It emphasizes the significance of the report by highlighting the data derived from the investigation that may or may not support the hypothesis made in the introduction, and often this summary is embedded in the discussion section or could be seen in the final segment of the abstract. Thank you for your endurance, for staying for the last session of the next to the last day of the meeting.